We are looking at details of energy generation in mitochondria and in chloroplasts. So we are going to start off by looking at mitochondrial structure and specifically then looking at oxidative phosphorylation that happens within the mitochondria. And then we are going to look at molecular mechanisms of electron transport chain. Um, and we will look at how those electrons are transferred from one molecule to the other, and how we use the proton pump as a way to generate the ATP to allow us to use that. And then we are going to talk about chloroplast and photosynthesis. And then finally, towards the end of the lecture, which is probably something we will see in the Canvas Studio lecture, we would look at how these systems were generated and how they evolved to be the way they are today, and if that is an efficient way for cells to work or not. There's something behind my laptop that is stopping me from moving it. Sorry about that. There we go. Now it's okay. So, like I said, that we have talked about the evolution of mitochondria and chloroplast before, all the way actually back in the first chapter, when we talked about how eukaryotes were generated over time through incorporation of actual prokaryotes, little bacteria that either had photosynthetic ability or that were able to utilize energy in a better way. And so those mitochondria and chloroplasts are those remnants of those bacteria. Some of these systems we know from looking at organisms like the electron transport chain evolved in the membranes a long time ago, over 3 billion years ago, in bacterial systems. So we know that that was where we first started to see it. And that is how it has gotten moved into eukaryotic systems as well. So in an electron transport chain, the membranes of that organelle and the mitochondria itself are extremely variable at each level. And they have specific, essentially, transmembrane proteins that act as a way for our electrons to be transferred, for our protons to be transferred, to create that gradient in order to allow for ATP generations. So these systems, they are usually, like I said, membrane bound. So all of the enzymes and all of the receptors and all of the proteins involved in this process are going to be bound to the membrane or associated with the other proteins that are bound to the membrane. And they are, three different ways that they can work. One of them is where we take the energy from food that has first been broken down into those pyruvates and acetyl CoAs, and then that can be taken into the cell, into the cell's mitochondria, where oxidative phosphorylation allows for transfer of energy from the activated carriers into the ATP and converting it into the ADP. Another thing that it usually is coupled with to create that ATP is a proton pump. And that proton pump basically uses the proton gradient to use those protons to bring them into the membrane, into the inner membrane space, from the intermembrane space into the actual core of the mitochondria to create the ADP. And then finally, you have pumps that can transfer protons across the membranes, as well as take in energy in photosynthetic organelles. So we will kind of go through each one of them more in detail throughout this lecture. So most of the energy that we are generating is getting generated through use of proton gradients across membrane spaces, whether it is from the outside of the cell to the inside, or whether it is inside the mitochondria, going from the intermembrane space into the actual core of the mitochondria. So in the first stage of electron transfer chain, you actually have these protons inside the mitochondria, and they are pumped across that membrane into the intermembrane space. As you take the NADH and FADH2 molecules and transfer those electrons from them. So those electrons, as they are transferred, they are going to result in protons getting pumped outside the inner space of the mitochondria. Then in the last step, where ATP synthase is working, the proton gradient that has been created by this pumping of protons into the intermembrane space, allows these protons to now be a lot more concentrated in the intermembrane space, and those are taken in by the ATP synthase. And the ATP synthase, this is a complex of proteins that is creating this intermembrane protein molecule that rotates. And as these protons are taken in, they allow for the energy from them allows for this phosphorylations of ADP to create the ATP molecule that we then use for energy consumption. So in the second stage, you take those protons and use them to create ATP. So essentially, you can think of this as a coupled reaction, just like we've talked about throughout in glycolysis and before as well. So the first part, where you're taking those activated carriers and you're ripping the electrons off of them and then using that to pump the protons out, that one is creating that store of protons in the intermembrane space that can then be used as an energy source for creation of this ATP in the second step. So cells essentially are harnessing the energy that they have gotten from those broken bonds in that initial glucose molecule or the pyruvates, and they are taking that in the form of electrons. And these electrons are what are providing the energy and giving power to the cell machinery that allows for it to then be used for our own energy consumption. So the energy from the electrons is converted to heat energy in a normal condition, but in our cells, it is going to be converted not just into heat energy, but also energy to work in the form of the ATPs. Now, this is not going to be as efficient a process, and so you will lose some energy as heat. Other is going to be stored as potential energy for our work in the form of ATP, and then you would have some water that is released as a result as well, or oxygen that is produced. So this is just showing you an example in real life scenario. If you had a battery that was just working by itself with a system, you would be converting everything to heat energy or to energy to do the work. However, if you have a pump that is transferring that energy to do multiple types of work, some of that energy would still be lost in the form of heat, while the remaining will be done to do the actual work that needs to be performed. In this case, to move the water from one reservoir to the other. Okay, so let's look at how this system works by first looking at where they are going to be working. So the two places that you're going to see these systems working is in the mitochondria or in the chloroplast. Both of these have one thing in common, in that they have a lot of different membrane spaces. So in the mitochondria, you actually have very, very complex matrix because of invagination of the membranes in and out of that space. And that creates this internal environment, which contains a lot of these type of processes happening, are happening inside that intermembrane, inside the matrix. And the space between the matrix and the outer membrane is what we call the intermembrane space. Now, inside the matrix is also where you will find ribosomes and DNA or any other machinery related to the mitochondrial daily work. Now, one of the, one of the proofs that we have so-called, or one of the reasons why we think that mitochondria and chloroplast came from a bacterial ancestor that was taken in by a eukaryote is the presence of this DNA, this circular DNA inside the mitochondria and chloroplast. These particular DNA, if you look at it, it actually contains a lot of different genes. But among those, it contains specifically genes related to the electron transport chain machinery. So all the different proteins that are needed to create that proper system to perform electron transport chain can be found on that mitochondrial DNA. You can also find ribosomal RNA genes on there that are specifically for the mitochondria in there. You can find the ATP synthase gene in there, but there are other genes as well that are present on that tiny little, circular piece of DNA. And some genetic disorders are actually happening because of mutations of these genes. Chloroplast as well. In the chloroplast, in photosynthetic organisms, you will also see the same kind of idea where you have multiple membranes. So you have an inner membrane and an outer membrane. And then inside the space, inner membrane is the space that has the complex machinery related to the energy production. And also, you also will see, again, circular DNA molecules that are specific to these chloroplasts that also contain similar types of genes on them as well. So, for this one, you should, again, note that there are inner and outer membranes in both mitochondria and chloroplast. They have their own circular DNA that contains important genes related to the work related to that particular organelle, specifically especially for electron transport chain. All the molecules related to, all the proteins related to the electron transport chain can be found, their genes can be found on the mitochondria itself. And also they have their own ribosomal RNA genes to create, they have their own little pocket of ribosomes to do the work that needs to be done to translate the proteins that are being made there. Okay, so we have again talked a little bit about that earlier as well. Because of these double membranes, that is one of the reasons we think that mitochondria and chloroplasts were formed through some kind of a symbiotic relationship between a larger cell and a prokaryote that it ingested. And when they ingested it, they were able to somehow survive in that space, and they were able to help essentially the organism that took them up, or the cell that took them up, by providing them now with photosynthetic ability or ability to more efficiently break down their own food and generate energy from there. So mitochondria and chloroplasts both have many features that are still, the same in modern day bacteria and archaea bacteria as well. They can divide on their own inside cells. They both have their own genomes, and they have their own replication, transcription, and translation machinery. So they are not dependent on the cell that they are in for that work that needs to be performed. Questions?
So that's super cool.
I know, isn't it? So yeah, it's kind of
like
little creatures inside. Yes.
In eukaryotic cells, during replication, the mitochondria follow the same directions that the cell is going, or it will
no,
replicate?
mitochondria and chloroplasts follow their own little cycle all by themselves. Actually, they do increase in number over the course of cell's life cycle, so that there are enough of them around to be duplicated, to be divided into the two cells as they are created, but they are not going to synthesize DNA when the cell is in S phase necessarily. They are synthesizing their own DNA when they need to replicate, and they are going to be dividing on their own. Now, mitochondria will be affected by the organism as a whole, and chloroplasts as well. So specifically mitochondria, you can have more mitochondria in your cell by exercising more, right? So there are studies that have looked at the number of mitochondria you can find in a particular, lung cell or in a particular type of cells, and they have noticed that the more exercise a person is doing, the more those mitochondria will increase, or intense exercise. It's not just like normal, typical exercise, but intense exercise. And that helps you build your stamina. So that is something that can happen. In certain populations, you will notice that there'll be more of mitochondria per cell than others. So that is one way to control it. However, they have their own generation system. They're always doing their own thing. Okay.
Okay, thank you.
Okay. So now mitochondria, like we said, are, obviously powerhouse of the cells. So that is why when we exercise intensely, you are going to increase over time the number of mitochondria, because the body's cells at that time will get the signals that, oh, we need more of these because we are doing more intense workout and we need more energy consumption. And that would lead to increase in mitochondrial content inside your cells. But you would see a lot of these throughout your cardiac muscles, organized in a particular way to help our heart muscle, contract and relax in that rhythmic manner. They are organized in between the myofibril contractile, structures in a very consistent way to create that rhythmic motion. And you can see that right here in this electron micrograph of cardiac muscle tissue, where you see the the contractile apparatus in between, and then you see these large mitochondrial structures, and you see how many of them are there in that space. If you look at a skin cell, you're not going to find so many of them lined up next to each other in there. They're not going to be as many of them there, and they're not going to be as organized in there. Similarly, in a sperm's tail, you would also see mitochondria lining the entire flagellar core structure, helping powerhouse that structure so that it can move in a very efficient manner, in a fast speed, to do the work that it needs to do, right? And these, like I said, these number and the way they're organized is different depending on what tissue you're looking at and what function it needs to help, actually power in that particular space. So in our other, like if we were to look at our general cells, you would notice that in our cells, they're also quite numerous in number, and they can be individual, just like a few clusters of them here and there, or in some cells, we will notice that they will be forming these elongated structures, which is basically continuous mitochondrion lined up against each other, similar to how you saw in a cardiac tissue or in another, in those form cells. So they can, when they are forming these structures, it is because they are fusing almost next to each other to make these long tubes where individual mitochondria have a continuous kind of intermembrane space to help us utilize that energy better and to help us harness more energy in there. Usually we see these continuous mitochondria close to the nuclei and around the endoplasmic reticulum, which is where a lot of the energy requirement is, which constantly needs energy production and utilization, both because of the high requirement of ATP inside the nucleus during transcription and during replication, and also due to the high requirement of ATP around the endoplasmic reticulum as the cells are translating the proteins and then modifying them for function. Inside the mitochondria, if we are to look at it a little bit more in detail, there are 4 separate kind of compartments. The outer membrane is your, initial membrane that is creating that outer space. It has that outer membrane is actually a little bit more permeable to many of the agents that are in the cytoplasmic space than a typical plasma membrane is. So this actually has a lot of transmembrane proteins and specific channels called porins that allow for more permeability and a more essentially signaling capacity between the cytoplasm and the mitochondria. Then inside there is a second membrane, which is your inner membrane. And that inner membrane is then invaginated and kind of folded to create a lot of these structures inside, and those structures are what are going to then create that matrix space that is going to be where all your electron transport chain and oxidative phosphorylation is going to be carried out. Now, in between the outer and the inner membrane, you have the intermembrane space. There's a space in between that is a little bit different in composition than the matrix space. This space contains, so these are all these little spaces. As those crystalli are forming, this membrane is getting the inner, inside membrane is folded up. It has this little space in between them. And that space is where, as you are coming in with your acetyl Coa molecules or other coenzymes, that's where ATP is going to be coming out of the matrix and going out, but also where those enzymes are coming in and the citric acid cycle is starting to happen. So that space is important in its own way. It is also where a lot of the cytochrome C and other apoptosis related enzymes are going to be released that then form the apoptosome and activate caspase 3 and do all that good stuff. So this intermembrane space actually has specific functions that are just specific to this space and that are important in their own way. Now, this inside part of the inner membrane, which is where the matrix is, that is where a lot of the actual processes required for oxidation of pyruvates and fatty acids during the citric acid cycle, those enzymes are going to be concentrated in this space. Also, the electron transport chain enzymes are all made there and are all organized there in that intermembrane, in the membrane itself, the inside membrane, along the matrix. And that's where a lot of those functions are going to be happening, right? Are going to be basically along that inside layer of the inner membrane that faces the matrix. I can't see my pointer. Can you guys see my pointer? Maybe I change it to an actual red pointer so I can see it easier.
I can see it every now and then.
Yeah, I know. That's what I was noticing because it's so white, the slide. No. Well, let's hit spotlight. There. Do you guys see that better?
Yeah.
Yes.
Yes.
Cool. Oh no, did I stop that? Spotlight. Okay. So yeah. So again, you have the outer membrane. The outer membrane is more permeable than a plasma membrane. It allows for a lot more contact and actual conversation between the cytoplasm and the mitochondria because it has more permeability due to presence of transmembrane proteins and specific channels called porins that allow for more things to come in and out of the mitochondria. You then have the inner membrane, the inside membrane that is invaginated to create those folded spaces. As it folds and forms these crista, these really thin channel kind of things, the inside of those channels is what you call the matrix. And in between these layers is what you call the intermembrane space. And the intermembrane space is very important in its own way It is the site where you have the enzymes that are going to allow for ATP to move out of the cell, out of the matrix and out into the cell cytoplasm. It's also where you're going to have a lot of the other proteins related to, as the pyruvate molecules and acetyl coenzyme move in, ready for the citric acid cycle. That's where a lot of those enzymes are coming in as well. But majority of the enzymes related to the citric acid cycle and electron transport chain are concentrated in the matrix, in the inside part of these membranes. And that's where a lot of those systems are working as well. Okay, so there. So the first thing that we will talk about is mitochondria and oxidative phosphorylation. So this is where we are going to, look at essentially the citric acid cycle, which is going to be required for all the different molecules that are coming in, whether they are coming from fats or polysaccharides. So for sugars, you go through, first breakdown of the sugars to create glucose, or changing of the sugars to create glucose molecules, which are then broken down in the cytoplasmic space, right? Inside the cytoplasm, to create, through glycolysis, to create the pyruvates. Those pyruvates then enter the mitochondria through these two membranes and into the space. As they are brought in, they are broken down to create acetyl Coa in that intermembrane space. So that by the time they get into the matrix, or into that matrix space, they're already an acetyl Coa enzyme associated with them. The fatty acids also from the fats are broken down into their monomers and to their individual fatty acid chains that are then brought into the mitochondria. And as they're brought in that inner membrane space, they start to break down further and they start to get modified to change into fatty acids that have acetyl CoA on them, that they get transferred from HS group. So that is your general requirement to get to the acetyl CoA enzyme before you can break it down to have that high ATP production. And so once you get the acetyl CoA, it's going to go through an oxidation process where you release a lot of these high energy electrons that are then going to be transferred to NADH and FADH, NAD and FAD, to create your activated carriers that can then get energy into the electron transport chain. There we go. So let's look at that oxidative phosphorylation. So the oxidative phosphorylation is linked with your electron transport chain. The first part that happens is that initial oxidative phosphorylation through the citric acid cycle and movement into that electron transport chain. This requires oxygen to be utilized by the cell in a very large quantity. So in the first part, you have your pyruvate molecules that are converted to acetyl CoA. In the process, you have your NADH from glycolysis that is utilized to activate this cycle, and then the electrons are transferred from there directly into the electron transport chain that we will talk about in a second. That also utilizes oxygen along with these electrons to then take those protons and shuttle them into the intermembrane space and produce water as a byproduct. This is the first stage of electron transport. So essentially the electrons are getting transferred from those activated carriers and getting utilized to shuttle the protons into the intermembrane space in this process. And then in the second step, you're going to have these protons that have been accumulated out into the intermembrane space, causing a proton gradient that will activate the ATP synthase. As these protons come in, they rotate that pump so that now the ADP can use that energy from that to create ATP by adding a phosphate group to this. So the way the electron transport chain gets the electrons from the NADH is here is your NADH molecule. It will have two high energy electrons that can be stripped away from that in this particular manner that it's gotten from the oxidation of the sugar that they were working with. And so these two protons are what is donated into the electron transport chain. In the process, the bond gets rearranged to produce your NAD plus molecule that is now ready to take in more electrons from the cytoplasmic space. And so the protons that are released from this hydride ions, you get a proton and two electrons from each hydride ion. Those two electrons get transferred into the electron transport chain, and the proton gets shuttled out into the intermembrane space. So each NADH gives off a single hydride ion that is then going to provide two electrons and one proton in the system. The electrons get passed to the electron transport chain, and the protons get shuttled to the outside intermembrane space. So here you can see that a little bit better. So you have your either the fatty acid chains or the pyruvates that are coming in from the cytosol. During the citric acid cycle, you're going to produce carbon dioxide, and you are going to release those electrons from NADH into the electron transport chain, and the protons that will be pumped out into the inner mitochondrial membrane space. And so by the time you get done with this process, you will then be utilizing those same protons back into the system and utilize oxygen and the electrons to create your water as a byproduct. Where did you go? Okay. So here you can see that in one of these cristae, how these systems would be organized. So you have your outer membrane and your inner membrane, and then you have one of these invaginations. On one side of this invagination, you see all the different steps that are going to be, there are 4 different steps in the electron transport chain that we'll talk about. So when the pyruvates and fatty acids come in, they get broken down into acetyl CoA. They go through the citric acid cycle and they release carbon dioxide as a byproduct. The NADH and the FADH2 that are coming off will get broken down so that you have the NAD plus ion, and then you have the 2 electrons that are shuttled into the electron transport chain, and the protons that are getting shuttled out. And so at each step, those protons are going to keep shuttling out until it converts the ATP, converts ADP into the ATP at the end of the day. So I actually don't like this diagram too much. I would feel better if they were kind of put off the other way, but it's okay. It just kind of confuses some students a little bit. But yes, so ATP is coming from the outside of the cell into the cell. The phosphates are going to be coming in from outside of the cell into the mitochondria. The protons are going to get shuttled back into the matrix when the concentration of them exceeds what's in there. So they move with the concentration gradient, and the ATP synthase with its rotation powers this reaction so that ADP can get that phosphate and generate ATP, which will then get shuttled back out of the cell. This entire process requires oxygen to be utilized, and water is produced as a byproduct in the process. Okay, so mitochondria at the end of the day is catalyzing a majority of this energy conversion, right? So that's where you're getting majority of your ATP. And it's taking essentially from the NADH, looking at just that, it's for each NADH, it's going to require half of an oxygen molecule. So 2 NADH would require oxygen, 1 oxygen molecule, plus 2 of these protons to create NAD plus and water as the byproduct. In the process, you are getting 1 molecule of ATP. So each NADH molecule can lead to 1 ATP production in this space. Questions about that? Yes, no?
I had a question about, so you said there's one NADH, but 2 protons needed for this?
Well, if you know, for each
Okay.
NADH, there's only 1 proton needed, but I was trying to equilibrate the equation. So I said 2 NADH would generate, require 1 oxygen molecule and 2 protons. Does that make sense?
Yes, thank you, just making sure.
Yes. In my head, half an oxygen never makes sense. So I always like mentally try to equilibrate, and I think I just said it that way. Okay. So let's look at now electron transport chain and how they are pumped. So for electron transport chain, there are several molecules that are needed, or in several proteins that are these that are embedded in the inner mitochondrial space that are needed to create that gradient, to create that chain reaction where you finally get your ATP. So it starts off with NADH dehydrogenase complex. So this is the one that is going to strip the 2 electrons away from the NADH molecule. The product would be that NAD plus that will go back out into the cytoplasm, and 2 protons, and a proton that is going to go out into the inner intermembrane space. So in that in-between space. It will then take in the second step, the ubiquinone as a product that will then be utilized by cytochrome C reductase complex to again shuttle more protons from the intermembrane, from the matrix into the intermembrane space, and release. This ubiquinone molecule gets broken down to create cytochrome C that is going to be released also in the intermembrane space. Now the cytochrome C, where else have you heard about that?
In the cell death.
Yes, in apoptosis. So what happens during cell death? What happens to cytochrome C?
It released to the cytoplasm by back and bags.
Exactly. So in apoptosis, the cytochrome C gets released outside, it gets shuttled outside the mitochondria into the cytoplasm. Now, yes, it's part of the apoptosome and does other things too, but what does that do to this whole machinery inside the mitochondria? What happens if cytochrome C gets thrown out? Can electron transport chain continue?
No, it will
No, right? It will inhibit that function. And what's more is that you still had a couple of steps where protons were getting pumped into the intermembrane space. So imagine that intermembrane space now, unable to bring those protons back in and unable to continue the electron transport chain properly, and instead just getting, over flooded with these protons. That's going to destroy its membrane's integrity. And that's going to be part of the way that apoptosis is going to start to happen. Does that make sense?
yes.
Okay, so the cytochrome C is then what goes into the last, into the third of these 4 enzymes. And this one is a cytochrome C oxidase complex, which will utilize the cytochrome C. And it is going to utilize, the 2 hydrogen, ions, and it's going to utilize oxygen to convert it into water. So in this one, we will actually look at that a little bit later. So in the first step here, the ubiquinone is your intermediate carrier for electrons. And then in here, the cytochrome C is the intermediate for electrons. So each time NADH, it can't just release these electrons and let them hang loose. It has to transfer them somewhere. So here you have your, ubiquinone molecule that is going to take in those electrons when the hydrogen ions are released or the protons are released into the intermembrane space. So the reason they can take that is because they have this hydrophobic hydrocarbon tail that is normally your oxidized ubiquinone. And then once the electrons are transferred to it, they get transferred along with protons. So you see this hydroxyl group on, oxygen bound to a hydrogen on both sides. That's your reduced ubiquinone. That's what's going to go in to the cytochrome C reductase complex. And inside, it's going to get broken down again to its oxidized state so that it gets the double bonds to the oxygen again. And those protons then get shuttled into the next step through here. So basically, the electrons can't just be released to transfer by themselves. They have to be transferred to a carrier molecule, either NADH or the ubiquinone or the cytochrome C, one or the other. Now, in some cases, you have metals that are tightly bound to proteins to help with that function. And that's the case in cytochrome C. So the cytochrome C by itself cannot be a very good electron carrier. But with the help of the iron ions that are embedded within it, they are able to bind both to the proteins and be a very versatile electron carrier themselves. So the cytochrome C doesn't work by itself, but with metal ions that are bound to it. So once those molecules have gotten those electrons and moved to the next step, in the very last step, you have water and oxygen that are being used up. And both of those are readily able to accept or donate electrons in the transport chain. So in that point, they can be taken up very easily without any help from intermediate steps. So the first step is your oxidized electron carrier, which is the ubiquinone. It's going to, take the electrons. You have the transient intermediate state. Then you have hydrogen ions from water that are going to reduce this electron carrier and put on that hydrogen bond to that. And then in the next step, you again do the same idea. So if you have a reduced electron carrier, again, you can create an intermediate state, which is positively charged. And in this case, the proton can be donated to water and lead you to an oxidized electron carrier. So throughout this electron transport chain, water becomes a very important mediator for these transports, which is again, why we must have 8 glasses of water, because we need the water throughout these steps in order for these steps to work efficiently. Now, the cytochrome C is one of the things that can also reduce oxygen in the system. And it does so by passing the electrons onto the oxygen in here. So you have the electrons that are donated by the cytochrome C, and that can go into your hemoglobin molecule or heme molecules and bind to the copper binding site. So again, it will use a metal ion or a metal carrier in this case as well. And that can allow for movement of oxygen throughout our bloodstream as well. Okay, so again, just like we've done before, the overall movement of ions in this case, and many times, is going with the gradient itself. It doesn't require energy input. It's either done through creation of the membrane potential or through movement of protons to create spaces that have higher concentration of ions either outside or inside the cell. So the electrochemical proton gradient that is established in that intermembrane space is a combination of that, the inner mitochondrial membrane having different type of potential on the outside versus the inside side. So on the intermembrane side versus the matrix side, along with that proton movement that is shuttled outside into the intermembrane space, allowing for them to be then shuttled back into the matrix to create the energy needed for ATP production. And so the membrane potential is done by proper orientation of membrane lipids, right? Just like we've talked about before, as well as the orientation of the embedded proteins that are going to be electron carriers at each step. So that orientation, if it was messed up, would mess up the gradient. And sometimes that happens in specific disorders where mutations would cause that orientation to be flipped. But normally you are taking in electrons from the electron carriers, you're taking protons from water, you're utilizing them together to release water, release water and transfer the electrons to the next electron carrier. Okay. So again, proton pumping is related, coupled with electron transport. So anytime you're transferring electron from one carrier to another, you're shuttling protons with the proton gradient either towards the intermembrane space or into the matrix. So when the concentration of the protons is low in the intermembrane space, each electron transport or each electron transfer will lead to release of protons into the intermembrane space. And when the the proton concentration is high in the intermembrane space, each electron transfer will lead to the movement of these protons into the matrix. So depending on how much proton concentration is present in the intermembrane space, the same carriers can either carry them in or out depending on what is needed. Questions from there? Yes, no, maybe. You guys are quiet again. There's one that I wanted to tell you about, but I don't remember. Okay. Yes, so the complexes are labeled 1 through 4. So 1 and 2 are together in that first NADH dehydrogenase. This one is complex 3 and this one is complex 4. And we'll talk about, you can refer to them that way if you would like. I'm okay with that as well. But normally I just talk about their functions. So that's what I'm going to be more focused on, is that what is the function of the first one versus the second one versus the third one. Okay. So this is your NADH dehydrogenase, and then you have your ubiquitinone that is taken in, and then you create your cytochrome C, and then you have the cytochrome C reductase. So, that's what I would like you to know them by. And then finally, at the very end, you have the ATP synthase. Okay, so in the ATP synthase, this is where all this hard work actually pays off. So in ATP synthase, you have a reversible coupling device. The protons, as they get higher concentration in the intermembrane space, start to get shuttled into the matrix through this ATP synthase. As they move through, they rotate the core of this ATP synthase molecule, and that basically provides the energy to the system so that the phosphate can be taken and phosphate and ADP can be taken into this enzymatic carrier and released as ATP. Now, sometimes you have higher proton concentrations inside the cell matrix instead of the intermembrane space. In that case, this same ATP synthase can actually work in reverse, where it can then utilize ATP that is present inside the cell or inside the mitochondria and release it back as ADP and phosphates as it shuttles those protons out into the intermembrane space. So it can work in both capacity depending on the needs for the cell. It is essentially like a motor, right? It's a rotor motor. So the top part, the part that is actually embedded into the membrane, is essentially a rotor ring. It has a lot of these alpha helices joined together in this ring-like structure that then the protons come in, they kind of rotate around it, causing it to rotate, which then allows the ADP and phosphate to come into the central stalk and bind to the ATPase head of that protein molecule, releasing the ATP as a result. So now in this process, now you can see how much energy is getting utilized and released at each step. So at NADH, you have a lot of potential energy. And at each step in this stepwise reaction, you're going to take some of that energy out of that system. And so from NADH to the NADH dehydrogenase, as you release that first proton and take those two electrons and put them onto the ubiquinone, you release a certain amount of energy that then gets further deteriorated as you go into the cytochrome C reductase complex, that then gets further reduced as you take the cytochrome C into the oxidase complex. And then finally, when you end up with your water release, you are back to the basal levels, or all that potential energy has been released at that time and converted into your ATP and the release of heat, which would be the lost energy. And again, this whole process is made possible because you have coupled transport, and you are able to take energy from one side to power the other reactions that would not go on otherwise. So the coupled transport at each step drives the electrochemical proton gradient, allows for those protons to be shuttled out so that they can allow more ATP to be generated. So each time a pyruvate molecule comes in, a proton is also coming into the matrix. And then each time the electron transport chain is moving, those protons are going back out. And so if you were to look at your mitochondrial space, the actual receptors and the actual transporters that are taking your pyruvates into the matrix are going to shuttle protons also along with them, right? So they are coupled transport here that is driving this proton gradient and moving these protons into the matrix. And then later on, when you are moving phosphates, they will also allow more ions to be moved in, more of these protons to be shuttled in. It's also a coupled gradient. And this is what is then producing those protons into the matrix, moving them into the matrix where they can then be utilized in the electron transport chain. Right? Otherwise, electron transport chain, every step pretty much is shuttling them out, except for the ATP synthase, which can go both ways. I click it once and it clicked twice. So here is the final count on your ATP production per single molecule of glucose. In glycolysis, if you remember, we produce 2 NADH molecules and 2 ATP molecules. Each one of these NADH, these were in the cytosol, can lead to 3 more ATP yield, but this is going to be slightly different because some of the NADH is impermeable to the mitochondria. The next one, the one that is in the cytosol, is partially impermeable to the mitochondrial matrix. The pyruvate, the next step is from pyruvate oxidation into acetyl Coa. Now you get 2 pyruvates in the end of your glycolysis. Each one of them is going to get broken down into 2 NADH in the mitochondrial matrix. So you're going to generate more ATP from them than you did with the cytosolic one. And so from these NADH, you would get a yield of 5 total glucose molecules. And then as you go through the citric acid cycle, you generate another 6 NADH molecules in the mitochondrial matrix. You generate 2 FADH2 and 2 GTP. Each one of these are going to yield a certain number of ATPs as well. So the GTPs will lead to, essentially one nucleotide is converting it into the other. You will get the same back. So instead of 2 GTPs, it will be converted to 2 ATPs. The FADH2 will lead to 3 ATP molecules, and your 6 NADH will lead to 15 more additional ATP molecules. And then the final total, once you get all of them together, would be your 30 ATP at the end of the day. That's your final yield. Now, questions on the respiration portion?
I didn't exactly understand why NADH in the cytosol will produce fewer ATPs.
So yes, so these NADH cannot pass into the mitochondria efficiently. So some of them are going to get lost because they're going to utilize their own energy to move them in. So that's why you only get 3 instead of 5.
Oh, okay.
Okay.
Yes, got it
So
now.
when the NADH is made inside the mitochondria, it's converted just like it normally would. But when it is outside in the cytosol, it has to be first shuttled into the mitochondria, and it isn't readily permeable, so it requires energy input.
And do we need to study this table? Are we going to have questions with numbers like that or no?
So what you should know is that the cytosolic NADH versus the mitochondrial NADH, why that number is different. You should know that the most energy is in the ATP, right? But that NADH produces more ATP molecules per NADH than FADH2, right? So the FADH2 is producing less energy overall than the NADH molecules. And the GTP can convert straight into ATP without that energy loss. Okay?
Okay, thank you.
And you should know that obviously the electron transport chain is where majority of this ATP is produced. So you should know the details of the electron transport chain for sure.
Okay, got it.
Okay. Okay, so if we were to add inhibitors of this process by adding what we would call uncoupling agents to it, that reduces the efficiency of this system, and it reduces the ability for the cells to produce as much ATP. So for example, if we add an uncoupling agent that can act as a proton pump and push those protons back into the matrix without ATP synthase input, then no ATP is going to be generated from this process, and only those that little packets of energy that was going to be created will be created. So you can actually inhibit the ATP production by using specific inhibitors of this process. Usually that involves exactly this, that you actually kind of have a competitive inhibitor to the ATP synthase that can take the protons and shuttle them themselves into the matrix. So we have done experiments in the labs where they have looked at specifically that to work out this process. In this case, we usually use, a artificial cell. So it's a sealed vesicle or a liposome to mimic a cell membrane or to mimic a particular type of membrane. We make these sealed liposomes either with nothing inside them. Maybe it's just a plain sealed liposome with no proteins inside, or sometimes we will add specific proteins in there. So in this case, they add the bacteriorhodopsin, which is a proton pump, into the sealed vesicle so that they acted as a proton pump in the membrane. They would orient themselves exactly like they would inside our body, inside our cells. And then when they are exposed to light, you would see the shuttling of protons from the outside environment into those liposome space. However, this alone is not going to give you any energy production. However, if you kind of incubate these liposomes with bacteriorhodopsin and ATP synthase, so that both of them can get incorporated into the membrane of this artificial cell, you then are able to generate ATP, because the proton pump will take the protons into the liposome, and the ATP synthase will utilize those protons to generate ATP. You obviously have to provide it with all the right environment, the material to create the ATP. So it should have ADP and phosphates available in there. But once you have that, it is going to work. Similarly, if you do the experiment with just the ATP synthase, you can have all the protons in the world outside, you can have all the ADP and phosphate available, but regardless of that, no ATP will get generated unless there is a actual pump taking the protons into this liposome so that they can be shuttled into the ATP synthase. So then in that very last part, you can see that you have both the bacteriorhodopsin and the ATP synthase, but now you've added an uncoupling agent, so another proton pump inside that space. And so while bacteriorhodopsin, when activated by light, will take protons and shuttle them into the liposome, the uncoupling agent is shuttling them out, so they don't have a chance to get to ATP synthase and produce energy. So that shows how ATP synthase alone was never enough to create energy. You needed all those, in between steps to allow for the electrons to get transferred to those intermediate carriers and shuttle the protons out into the intermembrane space so that they could be then utilized by the ATP synthase to power the reduction, the process of ATP production.